So a few of the facts that I find particularly interesting to share with people is the fact that fungi are not in any other kingdom but into their own group. Historically, they had been put into the kingdom plantae, but later has been determined that nope, they are not plants, but they are not animals either, and they have their own classification. In all the ecosystems around the world, they are the primary decomposers of cellulose. I would like for you to think about all the nutrients that plants and other producers are capturing either from the soil and from the air. And all of those get captured and jailed into these structures with a thick cell wall in some cases made out of cellulose. It is mostly fungi that had the ability to chemically break those cells and effectively releasing all those nutrients to make them available for the entire community wherever they are. Now, they are able to do this via external digestion. And I forgot to do my disclaimer, which is that I usually work with children, so you might find me a little more animated than what you're used to. This comes in place because to me, when I am talking about external digestion, I just see these microscopic organisms growing through the soil and go, excuse me, grow, 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 bleh, grow, 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 bleh, grow, 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 bleh. No, this soil is all mine. Don't you touch it, pesky bacteria. So effectively, secreting all those components and all those proteins, they exclude the colonization of other bacteria and other microorganisms via into their substrate until they are good and ready to let it go. These organisms have chitin cell walls, which is another very tough carbohydrate, a complex molecule that makes them sometimes difficult to penetrate. And this compound is also found in the exoskeleton of insects and shellfish. Now, 93% of all the fungal species in the world are undiscovered. So there is a vast wealth of knowledge out there for young and old researchers to get their hands into and begin to explore. Most of the fungi that we know and recognize as mushrooms have a really extensive fungal mycelium, or the body of a mushroom, that lives below ground. And this serves as a method of communication between plants and the fungi themselves. And we'll see a little bit more about this when we talk about that communication mystery. So, if I am right, and they are everywhere, the air, the soil, the water, inside you, why don't we know more about fungi? The reality is that fungi like to hide. As a human species, we are very much a visual species. And we have a saying to that effect: out of sight, out of mind. And if it's out of mind, is out of study. Fungi around us can be living inside of the soil, inside of the trees, plant tissues, human nails, lichens, other fungi, or any other organism. And in here, I have placed some of my favorite examples for you. In that upper corner, you'll be able to see a lichen photograph by one of my favorite photographers, Steve Oxford. This is a symbiotic relationship between a fungi, a photobiont or a cyanobacteria, and a yeast. And they live in perfect harmony, supplying each other with what their needs are. Not only can they do symbiotic relationships, but they might also be parasites, like this Rosella alomyces specimen. The border that you see here is the cell wall of a bigger fungus, which is called alomyces. This is an aquatic fungus that is very common in freshwater ecosystems. But inside, you see these amazing, intricate structures that are the spores of the Rosella alomyces fungal parasite. You also see more commonly in the forest floor mushrooms that are growing through the soil or on top of dead logs. But even in living plants, we get these endophytes or fungi that live in symbiotic association with plants, and they live inside of the tissues. Endophytes can be located through the body of the plant, and we will be able to see a bit more about that when we talk about that mystery. So they have been hiding, and to my rough estimation, they spend hiding 98% of their life. So we are not able to see them most of the time. Think about it for a second. If I go outside and I want to study the tree in my yard, I'll be able to study it today, or see it tomorrow, or see it next year for all we know. But fungi, I might see it today. If I was lucky, I saw it when it emerged, and I will see it tomorrow. But after that, I won't be able to see it at all for at least a year. Sometimes mushrooms do not fruit every year, and you will spend a few years, from anywhere from 2 to 7 years, waiting for it to fruit again. So they're quite mysterious in their own nature. Their taxonomy can be very baffling again, because these are organisms that have experienced convergent evolution and divergent evolution. With convergent evolution, what we mean is that species that are not related to each other have evolved the same mechanisms, therefore making them look very similar to each other. And by divergent evolution, we are referring to species that are effectively sister species, meaning very closely related, but they can look totally different. So morphology it's not really a good indication of how closely related they are. And these mushrooms have been hiding for ages. How much? Let's find out. So, one of the most amazing fossils of mushrooms that you can find out there is the fossil of Prototaxides. This fossil was thought to be living around 420 million years ago and 350 million years ago. And it was huge. If you see at the top of the picture, you're looking at one of the lead researchers of this mushroom, the paleobotanist Francis Huber, standing next to the fragment of this fossil. So this fossil was first discovered in 1859 in Quebec, Canada. When it was discovered, they said, oh yeah, something that big, something that tall, it has to be a conifer, it has to be a tree. Later, people continue to study this fossil and this organism, and they came up with the idea that it maybe was an algae because it doesn't have the same structure as a plant. So maybe. But it's not only 1919 that the first possibility of it being a mushroom starts to emerge. Even later, by 1961, it's not only Dr. Francis Huber joins the Smithsonian Natural History Museum that someone dedicates an amount of time, a significant amount of time, with the technology that allows it to say, wait a minute, this is no plant, this is definitely a mushroom. And he compared very thin slices of the fossil to the cell structure of polypores and mushrooms that are quite hard in nature. Through his work, we have the first vestige of certainty, or the first indications of any certainty that this is a mushroom. But it's not until we have the ability to do chemical analysis, isotope analysis into this fossil in 2007 that we're able to say this is definitely a fungus. That was quite the puzzle, and it was not solved until recently. Now, most of the rest of the fossils that are out there of fungi, and they are definitely very few and far in between, these fossils are microscopic in nature, either encased in amber or nicely preserved in the tissues of other plants. So we have this first fossil, the oldest fossil of fungi ever found. It belongs to Autospharia giraldi, and it was present 1 billion years ago. So it's effectively through the finding of these fossil that we are able to say, yes, fungi arrived first to land. They are the first land colonizers, and then they are able to form associations with aquatic and semi-aquatic plants and bring them in and aid them into colonization of land. Now, it is, these fossils are found in shallow water, very well preserved, so much so that you are able to see these chamber where all the spores of the fungus would have been. Now, the other specimen that I have here for you is the fossil of Savoreala genus. This is a genus that is still alive, and it has a structure very much similar to some plant pathogens that we are able to see today. We are able to see this particular specimen because of the silicification of its finding site, and it shows you this chamber with all the spores that the fungus would have produced, getting ready to reproduce. Now, this structure that we're able to see, it's called a perithecium, and it's like many sacs of spores encased into a bigger sac of spores. Now, this starts to give you some idea on to how far away the evolution of fungi began. So before the discovery last year of Autospharia giraldi, the fungi that were around 1 billion years ago were thought to be simple and single-celled organisms living only in water that exhibit asexual reproduction through zoospore. And zoospore is just a very big word to say that they have a motile spore, very much like what a sperm spore or a sperm cell would look like, where you have a globe or a sphere carrying the genetic material and a flagellum attached to the posterior cell, to the posterior side of the cell. So once their appearance 1 billion years ago, we get the first glimpses of that land colonization around 700 million years ago. The first ones that we know of belong to the kingdom, they belong to the group Blastocladiomycota, but as they continue their evolution, they lose the ability to move through water, lose that flagella, and find better ways to disperse. Those better ways are either by infecting other organisms, by becoming their symbionts, or alive in the forest floor for the most advanced of the species. So the phylum Ascomycota and the phylum Basidiomycota exhibit that very new, very modern group of species. And those are the phyla that if you go out to the forest or wherever you are, those are the macro fungi. Now, all of these species that we know represent around 144,000 species that have been described. They are skewed, of course, to all those species that we can see, but the estimation of the species is 2.2 to 3.8 million species, which is 6 times the number of species estimated for plants. Within those species that we don't know about yet are 16,000 that are called dark taxa, which that means is that is species that were detected from dust via DNA analysis, but no one has ever seen their specimen ever. So we know that they exist because we have their DNA, but nobody has ever seen them. These dark taxa can be anywhere. It can be inside of other hyphae like we saw. It can be inside of other insects or even inside our guts. To give you a little more information about how those species are organized, we have here in the top of this illustration the phylum Ascomycota. 90,000 of the species that we know today belong to this genus. And it has members that have saved our lives, like the mold Penicillium, which is the first organism that we were able to extract penicillin from. If you saw morels this spring, you saw a fungus in the Ascomycota phylum. Now, most of the time, it's not the Ascomycota that you're gonna notice, it's the Basidiomycota. And in the Basidiomycota, you have all those fungi that look like an umbrella and that many of us know and love. But you also have the yeasts. You have yeasts in Ascomycota and in Basidiomycota. You have rusts and you have the very fun jelly fungi. This spring, I also saw a lot of and also received many pictures of people finding wood ear jelly fungus, and that made me so happy. So keep your eye out every time you go to the forest, because I assure you there are many of my friends in there ready to say hi to you. By comparison, there are many of the phyla that we can't see with our naked eyes. Some are the fungi that attack your bread. Some are insect pathogens. Chytridiomycota are those that are responsible for the attack to the skin of amphibians. And we also have very obscure phyla like Blastocladiomycota and Cryptomycota. As these show, fungi are more closely related to animals than to plants. So that is one of the advances, one of the first advances that we were able to see once technology allowed science to move forward in the 60s. So we've been talking about them for quite a while now, but what are they? Well, they're fungi. Mushrooms are fungi, but not all mushrooms are fungi, but not all fungi are mushrooms. And some of the fungi that we know in our daily lives do not produce a mushroom. An example would be the molds, the yeast, or the leaf spots that you find in plants and trees outside. These mushrooms are the reproductive structures of the individual and are very much like apples to the tree. The apple is what you can see, the mushroom on the ground, and the tree would be that mycelium or that body of the fungus that is hidden either in the soil or in the wood or inside of the insect. Now, this is a point of pride for me because the largest organism in the world, it's not a tree, it's not a sequoia, it's not a whale, it is a fungus. And it was discovered via aerial surveys of the Malheur National Forest in eastern Oregon. This mushroom was none other than the honey mushroom Armillaria solidites, and it is 3.7 square miles in diameter. It has been growing for more than 2,000 years. So for those who are Christian, to put in perspective, think about Jesus was walking around, honey mushroom was growing. Same time reference. Now, they have been hiding for so long, but how do they do it? Well, let's start with that mushroom that we find in the soil. In the bottom side of that cap or the hat of the mushroom is the spore producing surface that is lined with thousands upon thousands of spores. When the mushroom is good and ready, those spores are going to be released and fly away with the wind. They can fly anywhere from a couple centimeters to thousands of miles if they catch a ride into an atmospheric phenomena like a Saharan dust storm or a hurricane. Once that spore gets to where they are going, it's going to germinate into a single hyphae. A hyphae is one cell thick and composed of a line of cells. It's very thin filament. Once that hyphae continues its growth, it will form a group of hyphae, which is called a mycelium. And that mycelium is what constitutes the body of a fungus. That mycelium will continue growing in the forest floor until it receives the environmental cue. Hello! Hello! It is time to reproduce! Winter is coming! When that happens, that mycelium will congregate and form the structure that we see and recognize as a mushroom, and the whole life cycle will start again. Since they are everywhere, the forest floor, other organisms, other fungi, they perform many roles in their ecosystem. Some of them will be plant decomposers. Like we mentioned, if they are not opening those cell walls, there are very few organisms that will be able to survive. Only a handful of bacteria have the ability to break apart those cell walls. They are also plant symbionts, and through the body of the plant, they make associations and they establish relationships with all plant parts. They are also present in the guts of ruminants, leafcutter ants, termites, you name it. If it eats a plant, it needs a fungal friend to capture those nutrients. But they might not always behave, and sometimes they cause disease, either plants or animals. But think about it. Any ecosystem needs a way to keep the checks and balances and to keep everything in balance. Fungi fulfill, pathogenic fungi for that matter, fulfill the role of keeping that ecosystem balanced. And if you ever have enjoyed a fermented beverage, thank a fungus. Now, in the forest floor, they perform one of the most amazing works that you can see, and is being a method of communication between plants and trees. This has been effectively called as the wood wide web, and I would like to show you a video about it. Oops.
Trees may look like solitary individuals, but the ground beneath our feet tells a different story. Trees are secretly talking, trading, and waging war on one another. They do this using a network of fungi that grow around and inside their roots. The fungi provide the trees with nutrients, and in return they receive sugars. But scientists have found this connection runs far deeper than first thought. By plugging into the fungal network, trees can share resources with each other. The system has been nicknamed the Wood Wide Web. It's thought that older trees, fondly known as mother trees, use this fungal network to supply shaded seedlings with sugars, giving them a better chance of survival. Those trees that are sick or dying may dump their resources into the network, which might then be used by healthier neighbours. Plants also use fungi to send messages to one another. If they're attacked, they can release chemical signals through their roots, which can warn their neighbours to raise their defences. But like our internet, the Wood Wide Web has its dark side too. Some orchids hack the system to steal resources from nearby trees, and other species, like the black walnut, spread toxic chemicals through the network to sabotage their rivals. Arboreal cybercrime aside, scientists are still debating why plants seem to behave in such an altruistic way. The hidden network creates a thriving community between individuals. When you're next in woodland, you might like to think of trees as part of a big superorganism, chatting and swapping information and food under your feet.
All right. So I would like to take this opportunity to see if we have any questions in the chat.
We did have a question. What did the first fungus use for nutrition?
That's a very good question. So the first fungi were so successful because they were able to extract nutrition directly from the rock. So they were, and this is a function that some fungi that associate with lichens still do today. So they release their chemicals into the rock and they break it apart. So they aid the weathering process of the mother rock.
That's the only question in the chat for now.
Awesome.
Yeah.
So let's continue our voyage. So this relationship with the roots of plants, it's called mycorrhizing. Myco means fungus in Greek, and rhizae relates to the roots of plants. This is a symbiosis that has been going on since 450,000 million years ago. Now, in this relationship, we know that plants usually produce much more sugars than are needed, but they require a lot of water and a lot of nutrients that they are not able to obtain. It's when they see themselves in need of especially nitrogen and phosphate that they open up their cells to fungi for colonization. They open up the cell wall, but they never open the cell membrane. So fungi are able to either crawl in between cells or inside of the cell wall, in between the cell wall and the same cell membrane. If your plant, let's say we have a plant in your garden and you're supplying it with fertilizer, the plant is gonna say, oh, no, I'm good at it, dandy. I don't need to open myself to attack from anyone. I'm gonna stay shut and you go bye-bye. So when you're adding these fertilizers that come out commercially, you have to keep in mind that it will reduce the biodiversity of those microorganisms in the soil. But when you allow the soil to have all its diversity, this really great relationship will develop in a way that fungi are able to exponentially increase the access of water and nutrients, not only by increasing the area, but also increasing the depth at which the plant is able to obtain nutrients in those soil particles. Now, they not only send water and minerals to the plant, but they also offer protection against desiccation and other pathogens that might be coming to the roots for a meal. Remember that this is mine, this is mine? That is the method that they offer protection. They are creating a chemical boundary around those roots, but they're also creating a physical boundary that pathogens need to go through in order to access them. Now, 90% of all plants in the world make mycorrhizae, but 100% of plants will make associations with fungi, because their associations are not relegated only to the roots, but they can happen through the entire body of the plant. And these associations can be between different populations of fungi. So we can have one population living in the branches, one population living in the stem, and another entire population living at the roots. There's another mystery that I love sharing with the kiddos because it blows their mind, and is the association that they have with as pathogens of insects. These zombies or entomopathogenic fungi attack and parasitize the body of insects. They're able to colonize the body, penetrate, and consume the body of the insect from the inside out. Once the fungus attacks the brain, then the insect would feel compelled to crawl up into a tree or a blade of grass or something that is higher up than the ground and clamp itself down or hug the plant tissue with the legs. Once there, the fungus is ready to spread its spores and infect more individuals. After this talk, this slide will be available for you, and I have placed a little video right here so you can go and watch how this happens in the case of carpenter ants. We also have bioluminescent fungi out there. What? I see these as neon signs in the forest floor. And the first person to notice this phenomena was Aristotle. He notices and documents this cold greenish glow that was present in damp logs on the forest floor. He called this phenomena foxfire, but it's also being referred to as fairy fire. And this is the product of the combination of luciferase and luciferin, two different proteins, into the mycelia of fungi. Now, this happens throughout the year for fungi that live in tropical regions. In our region, it is best observed during the fall on the ground in chunks of rotting wood, especially in areas with low luminic pollution. We have around the world around 80 species of bioluminescent mushrooms that scientists know about. There might be much more out there, as we already discussed. But most of the ones that are known have this umbrella-like appearance, which puts them in the Agaromycetes clade. Some of these mushrooms are able to glow their gills, others will glow the mycelium, but they will not glow all of their structures together. And I would like to share a little video about that glow.
Serenella, what constitutes a macrofungi?
Very good question. So a macrofungus is those that we can see with our naked eyes. Microfungi are those that we need to see with the aid of a microscope. So let me go quickly and share that video.
Now we're not sure why the fungi bioluminesce. And that's what we're trying to do right now, is figure out why. So organisms bioluminesce for various reasons, but it's probably all started out from just basic detoxifying processes that happen in the cells. And some of the antioxidants, as they break down, give off light. As these traits evolve, other organisms go on to gain additional functions, like fireflies use their light system for mating and communication between species. A lot of that also happens inside the marine environment. But right now, we're not exactly clear as to why mushrooms do it. The experiment that I'm performing out in the field is an ecology experiment where we are trying to understand the interaction of the mushrooms with insects. And so to do this, we're using fake acrylic mushrooms that we've produced in the lab and putting them out in the field covered in glue. Some of them have an LED light that has about the same wavelength as the mushrooms, so it's producing the same colored light. And so we're comparing the amount and the type of insects that are on the dark traps to the light traps. And by looking at the two sets of data, we can tease apart, which insects are attracted to the light and which insects are repelled by the light and which insects just don't care about the light. Through this, we can better understand the interaction of these mushrooms with their environment. It's very likely that the bioluminescence from the mushrooms has some ecological significance in the forest. Based off of our early results or early data, it definitely looks like insects are attracted to the mushrooms. Now, what role those insects are doing requires further study. Fungi are at this special moment right now where the research is really beginning to progress, and we're likely to figure out the mechanism within the next few years.
So that video was published a few years ago, and if YouTube would stop, I would love it. So that was published a few years ago. There have been several publications that state that fungi that glow, especially those that glow the mushroom part or the fruiting body part, are effectively calling the invertebrates to say, hey, come, I'm here, I'm delicious, come get a meal, and by the way, take my baby away. Could you please put my baby away? So it's for the mushroom part, it is postulated that it's a dispersal mechanism, very much like plants will use pollinators to disperse their pollen or their seeds. And
I have another question, Serenella.
yes, ma'am.
What accounts for the different timing of flowering? Some in the fall, some in the spring.
Very good question. So you have different fruiting times because of the structures or the strategies that they have to live. Those that are dependent on plants will have a really big moment during the fall to reproduce, because it's the end of the growing period where that sugar production is slowing down, that water availability is becoming scarcer, and the temperatures are lowering. Now, saprobes, or fungi that depend on dead plant material, will fruit through spring through fall because of the temperatures. During winter, it's very difficult to find a wide variety of fungi because of those cold temperatures that slow the growth of all species. Thank you for your question. Now, let me share a couple more slides with you. So in this presentation, I have also placed the do's and don'ts of mushrooms. So if you feel inspired to go out and explore, please make sure that you pick mature specimens only to give that mushroom time to spread its seed or spread its spore. Observe all characteristics that you're able to perceive: smell, texture, color. And if you're going to capture those individuals to bring them home and compare with some guides or the internet, make sure that you use wax paper or lunch paper bags to allow airflow. If you put a mushroom in a plastic bag, it will turn to unusable mush and you will not have a good time. So allow airflow to increase the longevity of that fruiting body. Please, 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 I cannot say this enough. Do not consume anything that you do not know. There is not any rule that tells you what's edible and what it's not. So you have to know the species before you're able to consume it. One of my favorite methods for identification and catalog of all the species that I have seen is iNaturalist. An iNaturalist allows you to take pictures with your phone. It's a free app available for both iPhone and Android users, and it will capture some of the data for you. So if you take a picture, it will get from your picture data the date and the location, and then it will use the picture to find whatever species looks most likely to be what you're observing. I am really glad that this software exists, not only because of the software, but because of the richness of all the experts and the community that interacts with the app and helps you identify and narrow down your identifications much more than AI ever could. So if you are doing this, not only taking the picture, but also taking the specimen, it's really good to do a spore print to also use that as one of the characters for ID. I have placed some resources here for your enjoyment. Once again, this will be available to you via email if you have registered with us. If not, please send me an email and I will send it directly to you. Thank you so much for spending some time with us and allowing me to play a little bit. And I think we have a few minutes for any last questions that you may have. Thank you again.
If you have a question, oh wait, here's one. Can you tell us a bit about the fungi that seem almost intelligent, like the slime molds?
Yes, so that is a great example because slime molds, first of all, slime molds are fungi lowercase because in the phylogenetic world, they are fungi-like, but not inside of the kingdom fungi. They spend most of their time as amoebas in the environment and not really as filamentous structures like fungi would. So what do slime molds use? Well, it's all about chemical communication. Those cells, they're single cells that will be out there trying to find nutrients and a way to communicate. So through chemical signals, they're able to detect the path that is closer to what they're looking for. Sort of what that advertisement said long time ago, follow your nose. So they're following their cell membrane noses towards the source of the food. And this is an evolutive trait because you need to be very efficient in order to find the shortest distance to your food. Otherwise, you would die.
And I think our last question, Serenella, this is a good one. I've heard about spore prints. You talked about it. How do you do those?
Yes. So a spore print is just the sign, if you may, that spores do when they fall into a surface. So if you take the cap of a mushroom, for example, and you break apart the cap, so stem in one hand, cap on the other, and put the cap down with the gills down on top of a paper. And I like it best when it's a white and black paper. So you have half of the cap on the white and half of the cap on the black for contrast. Once you do that, put a cup or a container over that to keep the relative humidity of the air really high, and that will activate the spore release. Give it anywhere between 15 minutes to an hour or two, and take that cap off, and you'll be able to see that design printed into the paper. So this is a way that you can effectively see the color of the spores of the mushroom that you're examining, and it's one of the characters that you can use for identification.